Welcome everybody to the statewide frost protection webinar. For those that don't know me, I'm Mike Basedow and I'm a tree fruit specialist with the Eastern New York Commercial Horticulture Program, part of Cooperative Extension here in the Champlain Valley. And today I'm also joined with a number of co-hosts from my colleagues throughout the extension and university system. We're really excited to have a great group of speakers today. Certainly with the last few weeks being a bit on the warmer side and with things starting to push a little bit early this season, a number of us have certainly been concerned about what the weather is going to be like for the next couple of weeks and how this might affect our overall crop development. Obviously, we still got a ways to go towards bloom. So we wanted to be proactive this year and just get together a group of people and have a good conversation about what options are available to us as far as protecting ourselves from any frost events that might come up. And of course, also hearing from our climate experts about where they see the next few weeks going for us. So we are certainly hoping to have a really good conversation today and hear from a number of speakers. So before we jump into it, just a couple of reminders for folks. We do have quite a group of people on today. So I do ask that you do keep your video off and your microphone muted throughout the presentations. I think between presentations, though, we'll have an opportunity to get all of your questions answered. And I anticipate, too, that following all the presentations today, we'll be able to open things up and really get your questions answered and have a conversation with everybody that's speaking today. So we do have 90 minutes planned. Again, we'll probably have some time for questions at the conclusion of the meeting, too. And again, as a reminder, it is all being recorded, so we will have this available to you all in the next couple of days. So, Mario, is there anything that you wanted to add in addition before we get rolling?
No, I really would like to encourage everybody to ask questions in the chat. We progress with the speakers and have a very open discussion, hopefully at the end of this webinar. Remember, we have 90 minutes, and perhaps we can even stay a little longer if we have a good conversation at the end today. That's it.
All right, great. Thank you, Mario. So I think with that, I'm going to go ahead and turn things over to our first speaker. First, we're going to hear from Jessica Spaccio, who's going to give us an overview of the weather that we've been seeing. She is with the Earth and Atmospheric Science Department as an Extension Support Specialist. And Jessica, I'll go ahead and turn things over to you.
All right, thank you. So welcome everyone. So yes, I am Jessica Spaccio. I'm a climatologist at the Northeast Regional Climate Center, and we are on the Cornell University campus. So I'm in Ithaca, New York. So I'll be giving kind of an overview of the climate monitoring that we do and how it pertains to apple bud development and freeze risk. So here at the Northeast Center, we do a lot of kind of tool development. We partner with different people, so ag is a big part of what we do as well. So we've created different tools, and then I'll also show some forecasts and how those play out together. It also have links on the page to the different resources that I'll be showing, and then on the end page as well. So everything that I'll be showing today is free and accessible to everyone. So you can feel free to take a look at these pages at any time. So we have an apple frost risk page, and on that page we have a few different resources. So here is showing an accumulated chill map, which isn't too exciting this time of year because we've exceeded chill everywhere. That's not too hard to get to. A lot of trees just require 1,000 to 1,200 chill values before growing degree days can start accumulating. So everyone's already met their chill requirement, but did want to show that that is kind of part of the calculation that goes into these seasonal forecasts, is that we have to meet that chill requirement before we start accumulating the growing degree days. So the next map here shows accumulated growing degree days at the point after which those chills were met. These are for Macintosh apples. So it's showing how many growing degree days have been accumulated throughout the region. This is a base 43 growing degree day that we use for that. So this is just showing what those accumulations are. So based on that, we come up with these phenological stages based on the growing degree days calculated and some studies that were done on when the stages would be met, how many growing degrees they need to meet these stages. So this map is showing that there's still a lot of dormancy in the tan colors. There's some green tips in the green, and then we've got the silver tip kind of in that silver color. Now this is based, like I said, only on those growing degree day accumulations, and the study of what kind of growing degrees we need for each of these stages. I actually heard some feedback earlier this week from someone who works with growers saying, hey, we're actually further along than your map is showing. And that may be that, our algorithm doesn't fit, the orchard that you're looking at, so maybe you're farther ahead. But all is not lost on our website. So let's say, where you are, which you will, right? You guys know your orchard's best. So what we have on the map as well is, so it's kind of drop down or different selections on the right there, but you can choose what stage you're already in. So you can say, well, I know I'm already in green tip, even though it says I'm not. So you can select the green tip on the right. And what these give is 3 different maps showing so each map is a percent of kill. So a 10% blossom kill, 50% blossom kill, or the 90% blossom kill. And this is based on green tip and for April 12th through June 30th. And this is just historical data. So this is not including any kind of forecast, but based on historical data, what's the probability that there will be a kill? So let's say you're in green tip and you're looking at that 50% map on the right. So it's green, so less than 10% probability based on historical data that there would be 50% blossom kill. So here I kind of give some more examples. So here's green tip at 10%, just so you can see a little bit larger. So it's based on historical data and a probability that would occur. So you can see in this map, there's more of those yellows. So a 10 to 15% probability, 50% probability in those yellow areas. And then another example, like say, well, actually I'm past that, I'm at tight cluster, or I'm almost at tight cluster. So you could look at that map. And here I've just shown the 50%, but again, at that main page, you can look at those 3 different probabilities, 10, 50, or not probabilities, but percentages of clusters. So here's the 50% blossom kill. If you're at tight cluster, and you can see there's a broader area that's in yellow. So meaning that there's 10 to 50% probability that 50% of the blossoms would be killed, again, based on historical data. Another tool that's super helpful is the Climate Smart Farming page. Hopefully many of you are familiar with this page, but there is actually an apple stage and freeze damage probability tool. So this is really useful, and this I'll kind of go through and explain. There's a few things we're looking at here. If you look at the orange map on the bottom is the temperature needed for 50% damage based on the estimated stage that you're in. And then this bright pink color is the daily minimum temperature. So the solid lines are on, and I've just chose here Penn Yan. You can choose any location in New York, which is really cool. You can go to change location, you can put in your location, or you can click it on the map, say, oh, I've got an orchard here, click on it, it'll give you data for your location. So again, so this is for Penn Yan, and this is for Empire. There's also 3 different apple choices. So the pink shows that what the minimum temperatures, observed temperatures have been. So this is through yesterday. So the minimum temperature was a little over 50 yesterday. And then these dotted lines are forecast. So it does take in a 5-day forecast. You can see the big jump here in the damage temperature because it went from dormant to silver tip. And so the temperature needed for 50% damage now is at -25. It's, I don't know, probably something like 10. But then you can actually click on it. Obviously, these are screenshots, but in the live, you can click on it, you can look at all the stats, and what you get is this little pop-up map that I've shown. So here I was hovering over April 17th. So this is showing that the forecast is for a min temperature of 50. And at that temperature and at the forecasted stage of green tip, the temperatures that you would need to get those different damage levels. So for the 50% damage, if you get down to 10 degrees, you'd be expected to lose 50% of the green tip crop. But then there's also 90% damage if you got down to 4, if it got really cold. If you're only at 19 degrees, only expect about 10% of damage. And so this is really important. Just obviously the forecast is helpful in knowing, and basically what we've seen is as climate is changing, we're seeing our growing season, our winters are becoming more mild, right? So we're getting this, we're growing earlier, right? We've seen more, the apples are coming out earlier, green tip is being earlier, the blossoms are coming out earlier. So with climate change, we're seeing those come earlier, but we're still at risk for freeze and frost, which is the real challenge here, that these, the temperature increase is outpacing still the risk of having that frost. So even though we're warming and we're getting those warm temperatures, the frost risk is still there. And so this is trying to like help you make those decisions and see what is coming. Here's just another example of several years ago. I guess it's more than several now. I remember it like it wasn't long ago, but 2012. Many of you are very familiar with the really bad losses of the apple crops that year. So it was a little bit similar to this year. We had a really warm early spring. March was really warm, but it was, so things came out, they blossomed, but it was still too early to be beyond the freeze risk. So you can see on the map, April 19th, that temperature dropped way down. You see that pink line go down. Minimum temperature of 16 degrees was hit, which is just below the 50% damage temperature of 17. So that's when a lot of that crop was lost that year. Also on the Climate Smart Farming page is the growing degree calculator. So if you're maybe less interested in, the other tool, this you can just look at the growing degree days. And this I did base 43. This is example for Rochester. So on this map, there's a few things going on, but the green is the season to date observed. So this is the accumulation of growing degree days, base 43. And then these purple and blue lines are showing the average. So the 30-year normal or a 15-year average, if you want to look at that, you can see Rochester has been since, I guess, late February has been well above a normal with these growing degree day accumulations. We kind of got a little bit cooler there. You can see we got a little closer to the 15-year average, but still above normal. And then this week, temperatures have been warm again. So those growing degree days really shot right up there. And then this also does include a growing degree day forecast that was just shown in red. So I took a screenshot for the April 17th, the last day of this forecast. So at that point, expected to have 187 growing degree days in at that point, which is well above the 15 and 30-year normal. And then I also put in a line just to kind of give an idea, tight cluster, estimated around, I think, 138 growing degree days. So it looks like in Rochester would surpass that in the next day. We also showed another example, Plattsburgh, farther north. So the growing degree days are much less, but you can see still a big jump. The last couple of days they're jumped up, and then the forecast with these warm days kind of continuing over the next week or so, that the forecast does show more coming. And then islip I had done as well. You can see, since late February, growing degree days above normal, got a little bit closer here in early April when things cooled down a bit, the end of March and early into April. But then they've already passed, according to this calculation, the growing days needed for a tight cluster stage. But then it shows again the forecast, so you can see that those growing degree days are going to keep climbing. So if you want to use your own calculation for growing degree days, you can say, okay, well, I'm forecasted to be 240 growing degree days. That means I'm estimating that my orchard is going to be at this stage. And then you can go back to our risk maps to say, I'll be at this stage. What's the probability based on historical data that will have a frost risk? So that's a lot of historical data, a little bit of like a short-term forecast. I guess I've got another map to show here. I did just another location. This one I pulled a couple days ago, so it's not quite as up to date, but just showing again where you can see those growing degree days and how they've accumulated and how they're forecast to change over the next few days. So I wanted to show some more work with data to kind of give you an idea of what your frost risk is. This is at our a page that we create. It's Climate2. The link is there. And again, you can search for any location. You can type that in and look for any location. So here's a frost freeze summary. So this I did based on a 28 degree temperature, and basically showing when was the last spring frost, and it shows the value as well. And then I just kind of ordered them so we could see, for this one's for Rochester, when was the latest spring frost. So it was in 2020 actually, and it was May 13th. So frost can come as late as mid-May. So unfortunately, with these warm temperatures, we're not out of the woods for having frost. That risk is there. And you can see the other years when there's been these late temperatures. And then the bottom is nice. There's a summary. So the average date of last frost is April 15th. So we're not far from that. But things can go on either side of that, right? So there's an early March 18th was the minimum. But again, that latest being in May just shows that risk is still there. Did the same thing for Geneva. And again, you can look at any location. So their average is again around mid-April, but they've had it as late as May 13th. Plattsburgh up farther north, their average is April 22nd. But again, the latest they had was back in 1963 was May 12th. Oh, I did want to point out, you can see in these two, there's Geneva, the 2012 big freeze event there. So that occurred on April 28th in 2012. So that's one of the top ranking latest freezes. I also threw in Poughkeepsie. I know there's some orchards down there, again, kind of mid-April, but look, they had one back in 56, May 25th. That's pretty late, but it's possible, not out of the realm of possibility. Islip stays a little bit warmer. Their latest was April 20th. I also say that these are based on the period of records for each of these stations. Islip has a shorter period of record, so we don't have some of those earlier years. So that may be leading to this maximum being only in April, not a little farther. The data just isn't quite as long in that area. But just the idea that this can occur. Another calculation that I did to kind of give, okay, I know you guys are really interested in these last couple weeks of the month is, what is the likelihood of getting a frost during that time period? I know many of you listened to another webinar that went over forecasting for this period and for the spring and summer. And I'd really, I'm going to go through some other outlooks too, so you can see further ahead than just these 2 weeks. But really let you know that these forecasts are based on some of this, like this one I'm showing right now is historical data, but also going forward, we look at current trends. We do look at ENSO a bit. We look at climate change quite heavily, and sometimes that basically what we've seen in the past 30 years that things are warming, and that goes into these long-term outlooks as well. And so there's a lot of really scientific data that goes into these outlooks. Things that do not go into these outlooks are solar or lunar cycles. It is not something that a true scientific meteorologist puts into their forecast. And I think that's just worth noting that not everything, unfortunately, that gets put out there is very accurate. I'm not aware of any kind of 47-day cold period cycle. So I have nothing really to add to that. But I will look on that period, that kind of the third and fourth week of April, to give us an idea of what could happen historically before we get into a little bit of the outlooks. And, I've already shown kind of those last possible late frost risks. So we've had warm temperatures and there's frost risk. So I guess be prepared as you can that's what can happen. So it is based on the historical data. And again, these are different for the different stations, period of records, but I looked at these last 2 weeks of April and I said, when was there, how often the years did it get down to 28 or colder? So at Rochester, 41% of those years hit that 28-degree or colder. Geneva, 44. Plattsburgh 60, makes sense, they're farther north. Poughkeepsie 42 and Islip only 2. That seems pretty low, but again, I put a little caution because they have a shorter period of record. So based on historical data only, there are chance for a freeze during those last 2 weeks of the month. So let's look a little bit more based on, instead of just the historical data, but a little more predictive. The Climate Prediction Center puts a lot of work into their outlooks. You can go to their page and look at all these outlooks. You can also, if you're really into it, the author of each of these puts out what they use to make these outlooks and a discussion on what goes into these. So you can see exactly what they're basing their forecast on. So the 6 to 10 day outlook. So this would be April 17th to 21st. they are calling for 60 to 70% probability of above normal temperatures. So that's a pretty good likelihood that we're going to be warm during that period. But a little bit of caution, we're going to next look at the 8 to 14 day period, which does overlap a little bit with this. So that covers April 19th to 25th. So this map looks different, right? We've seen those above normal temperatures and we see a little blue on the map. So basically, it's just a slight favoring of below normal temperatures, 33 to 40. So it's slightly favoring below normal temperatures for those areas shaded in blue. We do see the normal favoring normal temperatures in those areas shaded in gray. So this is for, again, so it kind of overlaps the 6 to 10 and the 8 to 14. That is in that kind of those times of concern. So there's possibility for some below normal temperatures there. The 3 to 4 week outlook map comes out. This one isn't updated as often. I should say that both the 6 to 10 and the 8 to 14, those are updated every day around 3 o'clock in the afternoon. So it's really great. I'm glued to these every day. I'm checking out what they're saying. So you can look at these every day and see how that forecast is changing. The 3 to 4 week is a nice because it's a different time period, but this one isn't updated as often. So this one is actually was issued April 5th. So it's calling for above normal temperatures, but it's based on some older data. They also put out a seasonal outlook. So they have April, May, and June, and they actually have seasons going on for quite some time. So you can look at May, June, July, and et cetera. And they are calling for favoring above normal temperatures, 40 to 50% probabilities in those kind of the right half of the state. And then the western half, 50 to 60% probability of above normal temperatures. So they're not forecasting an exact temperature, just whether those temperatures would be above, below, or near normal. And I should also say, even the 8 to 14 day, that's for the entire period. That doesn't mean that there could be one really cold night and then the rest of the days are warm, and it kind of averages out. So, that's definitely a word of caution, because all it takes is one really cold night, like in 2012, to hurt the crops. So those are for a period. I know it's quite a bit of info, and I do have just on the back, I've got my email there, jlr98@cornell.edu. If you have any questions that we don't get to during the presentation today, the Climate Center, and then those lists of websites that I showed to you. Again, those are all available for anyone to go through and look at, free of charge, no login needed. Just go and check it out. And that is what I have for you guys today.
Thank you so much, Jessica. Really informative presentation and tool that I have not known about. So really interesting stuff. Just want to check the chat box and see if there are any questions for you. I do see one at the moment. Philip asked if there was a particular reason for the changing base temperature in the degree day calculations for some of the graphs that you showed. Any particular reason for that, or just what you happen to pull at that moment?
Yeah, I think most of them are actually for base 43, unless it's possible I'd pull, oh yeah, I guess there's 42. So what's really neat in that calculator, there was some different ones. So 43 is what we typically use for apples, based on just kind of what the NRCC has done. But when you go into that climate smart farming tool, you can choose any temperature. So which is the cool thing about the tool. So if you're like, at my farm, I really think I'd rather look at 42 than 43, you can put that in there.
Okay, great. Thank you. One more question that I had for you was, you mentioned on that first graph that you showed, or figure that you showed, you were able to change it to your actual bud stage development. For the graph that you showed on the next slide, would you be able to change it there too? I think it was that Penyon example where they might have said they were at green tip, but if you knew you were at half-inch green or tight cluster, could you change that on that slide?
Let me pull up the slides. I want to make sure I'm looking at the right one. Oh, are you seeing my whole screen now? Are you seeing
Nope, I'm seeing it on your desktop. Yep.
Okay. Well, can you see it well enough?
Yeah, that's fine.
Okay. So was it on these maps?
I think it was the next one that you showed. Yeah, that one.
Yeah. So yep, you can choose any location for these, but the stage you can't change. That gets automatically put in for you. There is some other you can change, like when you want the accumulations to start or your season to start. So there are some things you can change on that page.
Okay, great. Thank you. All right, I'm not seeing other questions, but if folks do have any more questions for Jessica, I'm sure she'll be, I shouldn't speak for her, but hopefully willing to stick around with us a bit longer and answer them in the chat box. So thank you again. And I think we're going to go ahead and introduce our next speaker. Next, we're going to hear from Dr. Jason Londo of Cornell AgriTech, and he's going to be telling us about the physiology of frost damage and some of the practical challenges of testing some of our different frost mitigation tools. So you're good to go, Jason.
All right. Look good to you?
Yep, looks good.
Okay. Well, thanks everybody for having me here. Yeah, this is going to be just a brief discussion on sort of like what frost and freeze do to plants and some of the complications with preventing that kind of damage. So to start out, I just wanted to define a couple different frost and freeze aspects. And the first is that there's different ways to kill plants with low temperature, and sort of the way that these weather and climate related events happen can really impact whether or not you have any chance of defending early spring phenology. And so the first is to define advective freezes and frosts. And these are defined by cold moving into a region. So these arrows would be indicating sort of a wind speed or something along those lines. And you get a temperature gradient of cold air blowing in from one direction. And one of the key characteristics from the plant's point of view is that because you have this cooling event coming through, the plant tissues and the air tissues quickly reach an equilibrium. So if you have a weather station out in your orchard and you have an advective freeze or frost coming along, and you look at the temperature on your weather station, that air temperature is very similar to the temperatures that the bud tissues or flower tissues are experiencing. That's different from a radiative frost, where you have very little or no airflow. And during this radiative frost typically happen when it's very still and when there's a very open sky. And what happens here is the cooling occurs from the ground upward. And here the soil and your plant tissues are actually losing their heat. And the process of losing their heat is what's causing the cold temperatures near the ground. And so here it's really key because the plant tissues are actually physically losing their heat. The tissue temperature is actually lower than what you would detect with a weather station. And so it's important to think about when we get freezes, what kind of freeze we get, because the actual experience that the plant is going through could be very different. During radiative frost, these are the type of events where things like frost fans can be somewhat effective at mitigating, and that's because they are mixing the warm and the cool portions of the air temperature and raising that temperature close to the ground and the temperature that the plant is experiencing. I'm sure most of you know about frost fans and how they work, but I wanted to include a real quick little video just so you could see the effectiveness of frost fans. And this is something that a visiting student from the Netherlands in my lab allowed me to show in this presentation. On this movie is a frost fan functioning to raise temperatures in the Netherlands. And you can kind of see how as that fan moves around in two different axes, it pulls warm air down from on high and mixes it up with the lower temperature air, and in general raises the temperature of the whole orchard. So radiative and advective freezes are very different. You can have events that are a combination of the both, but things like frost fans only work with radiative cooling events. If you have wind flow, it defeats the ability of the fan to mix temperatures. Oh, that started again. Sorry about that. All right, so now let's talk a little bit about what actually kills plant tissue. And we have to differentiate things a little bit here. There is chilling damage and there's freezing damage. And so chilling damage is typically what happens to things that plants that are tropical or subtropical, where they're not optimized to grow at low temperatures, but here the damage can occur even above freezing. The damage that occurs is typically associated with the fact that you still have bright lights but low temperature. And so if light is coming in and hitting your plant tissue and it's trying to actively photosynthesize, but the temperatures are very low, the photosynthetic machinery kicks off a lot of free radicals, and those free radicals can cause a lot of cellular damage. You can also have changes to the function of the membrane in that it gets stickier essentially, and that can lead to cellular malfunction. In the past, foliar sprays have been shown to mitigate this kind of chilling damage in some crops. And the primary way that they work is they scavenge those reactive oxygen species. This is sort of like taking your vitamins, right? If you can keep those reactive oxygen species from bouncing around and causing a lot of cellular damage, in general, the plant is healthier. That's not really the type of damage we're talking about when we talk about frost and freeze damage, particularly in apples. We're talking more about when water physically freezes within the cells. When you think about where ice can form on a plant, it can form both outside the cell, which is extracellular or apoplastic, and inside the cell, which is intracellular or symplastic. And I just include those scientific words in case you're reading up on this topic and you want to know how these things connect. The extracellular freezing, the freezing outside the cell, doesn't necessarily damage the living cell, but what it does cause is it causes a dehydration stress that helps pull water out of the portion of the cell that's alive. And pictured on the right here is my best attempt at drawing a plant cell for you. And so basically what happens when you have ice forming outside the cell, it doesn't necessarily damage the cell. And as that ice builds up, it can cause dehydration in the cell. And dehydration itself is not necessarily lethal. It depends on how far that dehydration gets. What happens when you dehydrate that interior part of the cell is you bring all your membranes in close together. All the different organelles and the plasma membrane come close together. And if you lose too much water, all those membranes can kind of stick together. And because they're all made out of fatty lipids, when the cell rehydrates, when the ice thaws, you could sort of rip yourself apart because those membranes don't remember who they belong to anymore. You can also have freezing inside the cell if there's still water within the living portion of the cell that hasn't been dehydrated out, and that freezing inside the cell can also cause mechanistic damage to the cell integrity itself. Some plants can suppress freezing inside the living portion of the cell by increasing their sugar or protein or nutrient levels, basically by changing the freezing point of water. But freezing point depression only works for a couple degrees of suppression, so near freezing. So it's not a defense that's going to prevent damage at very cold temperatures, just right around the freezing point. The other thing to keep in mind is that in spring, when you have bud break and floral development, that the vasculature that connects the stems, the branches, the trunk to the leaves and the developing flower is all connected. There's tubes of water there. And so freezing within one tissue can propagate to other tissues. So if you're going to try to prevent freeze damage to an organism that is as complex as a tree, you have to protect a lot of that organism to prevent ice from forming anywhere intracellularly. Currently, the best defense to frosts is frost fans and overhead sprinkling systems. And I am not an authority in either one of these. They're just the most effective way of altering freeze stress at the orchard level. And they work by modifying the temperature that the plant experiences. That being said, there's a lot of people who like to test products to modify particularly freeze point depression or reactive oxygen species scavenging. Me included. I'm testing these products on grapes this season as well. We're all looking for a silver bullet that's going to help us keep our plants safe. So there's different approaches. There's the nutrient-based foliar spray approach, and the predominant chemistries that make up these sprays are chemistries that involve nitrogen, potassium, or calcium. Reviewing the literature using nitrogen-based approaches is not usually a good idea because it often promotes growth, and the more advanced growth you are, the less freeze resistance you have in general. And that's simply because the more development you have in a plant, the more water you have in your cells. Potassium-based approaches is another way of trying to enhance freeze response. It has been shown to increase freeze resistance, but if you look at the fine print, it works when crops are deficient in potassium to begin with. And so the plants, if you have weak plants to start with, you can enhance their ability to survive a freeze by boosting their general health. If you look across systems, use of potassium tends to be inconsistent among herbaceous crops and woody crops. And I'm actually an author on a potassium test on grapevines. It worked sometimes to very small effect with certain cultivars. So it's very inconsistent. There have been a few reports that calcium has an effect, but it is unclear what the mechanism would be for that effect. And so because there's so few studies, it's really hard to say whether this has any more consistency than nitrogen or potassium. Another approach is spraying PGRs. And the most effective way of dodging frost and freeze is by suppressing growth. And so the PGRs that reduce growth and change phenology, things like ethafon or ABA, they work by keeping the bud less developed, keeping the plant tissue less developed, and so they survive lower temperatures. It's not really something you can do once you have flowers because you've already kind of advanced to a very sensitive freeze stage. There are other ways to sort of work around when you get frost damage, like using Promalin, and I know Terence is going to talk about that in a minute, so I'm not going to go into that. But the most effective use of PGR so far has been this growth reduction. There's also the use of bactericides, and the way that this works is you go out and you spray and kill ice nucleating bacteria. And this is a type of bacteria that occurs in nature, and these bacteria function, or they have the function of forming ice crystals at temperatures warmer than the tissue would normally freeze. And it's kind of a tricky concept, but imagine your tissue freezes at 27 Fahrenheit. Ice nucleating bacteria may make that tissue freeze at minus 29. And they do this just because they alter the way that ice crystals form. They work very well to seed ice crystals. And so one way to prevent frost and freeze is by killing all these bacteria off, allowing the plant to use its natural defenses. As you can imagine, this is tricky because you have to spray enough antibiotics to kill off bacteria, and you need to do it within a time frame that's close enough to the freeze that the bacterial population doesn't swing back around. I also want to show you some of the challenges of understanding how frost and freeze works. And so I'm going to show you some preliminary data that Judith's been doing in my group. This is, you can see there's a bunch of apple sticks here with buds and flower, some green tips, some tight clusters, some exposed flowers. And then what these wires are thermocouples with little needles in it, and we can measure the temperature inside each tissue. And I'm going to show you a thermography approach to understanding when these tissues freeze in the next slide. And this is the easier way to see the distribution of what you're going to see in black and white. Okay, so this is a insulated cooler. It goes into a big programmable freezer, and then we slowly freeze the tissue, and we use thermal vision and these thermocouples to measure the freeze points. So that's an example here in black and white. So now we're looking at these different apple twigs with buds and open flowers. And what you're going to see as I play this is that you're going to see the different stems and the different tissues light up as like a brighter white, and that is the freeze event. If you're paying attention, there's a lapse time at the top here and the air temperature in Celsius. For the whole video, I can give you the summary stats. We go from 23 or 29.3 Fahrenheit down to 23.7 Fahrenheit over 36 minutes in this video. And what I want to do, just show you here, is that freezing occurs from top to bottom across these different apple twigs. It also freezes from the woody parts to the leaves and flowers, and from the leaves and flowers back to the wood. And in particular, I want you to see this one in the background here is going to be the last one to freeze towards the end of the video. And you'll see that when it freezes here at the base, it freezes up the stem all the way into the open flower. And so even if you're protecting the floral tissue from ice exposure, freezing in the stem tissue leads to freezing in the floral tissue. All right, so this is just to show you that freezing occurs over a really large range in different tissues, and the freezing events can happen in different directions depending on where the ice first forms. Here's another example. These are from the needle thermocouples that are inside the different plant organs. And this is far too complex to look at in this talk, but I'm sure we'll be distributing PDFs of this so that you can look at it later. What we're looking at here is 3 different cultivars and 3 different tissue types. And the diagonal line here is the gradually decreasing freeze air temperature. And when you see these lines spike up from that baseline, Those are the freeze events for those different tissues. And I've summarized it on the right here. In the blue bracket is the range of temperatures at which the bud freezes. But interestingly, though we can see the bud freezing, we can measure it freezing, we can see it with thermography, the buds will actually not die. If you put these back in water, they'll push and be fine. But as you get a little colder, you'll see the temperature where the leaves start to freeze. And in this case, it's a lethal freeze. And also where floral tissue starts to freeze and die. Right? So again, the take home point here is freezing occurs over lots of different temperatures. It's not wholly random, but it has the appearance of random. And this leads to some of what you probably observe in the field when we have a frost or freeze. When you have parts of trees that show a lot of damage and parts of trees that don't, there's an element of microclimate, but there's also the element of tissue to tissue differences in freeze events. So just to sort of wrap up this, testing for frost resistance. So my personal opinion, which is scientifically informed, but it is my personal opinion, is that foliar applied products have been studied since the 1960s for suppressing freezing and preventing frost damage. And in my opinion, we still don't have a workable product. Modifying the internal metabolism of plants, particularly in the spring when there's not a lot of extra heat for metabolism, it's unlikely to work unless the plant was weak to start with. The best options are fans or sprinklers, or perhaps some of the insulative products that I think Brent is going to talk about later. All of those work on modifying the temperature that the plant experiences, not necessarily buffering its ability to survive freezing. If you're going to test products, you really have to leave unsprayed controls in the blocks at which you're testing. I totally get the complication of spraying a product that you're hoping is going to prevent frost and then leaving certain parts of that block exposed. But if you don't have within block control versus spray groups, it's impossible to tell if there isn't another factor that's affecting the way that your plant responds to that event. And I also want to put out there, and I'm happy to coordinate with any growers who are testing products to try to evaluate product effectiveness. The key, though, is having some way to differentiate the control from the spray within the same block structure. And that's all I have for you today. I just want to say thanks again, and I'm happy to take questions. I put my email address up on here if you want to email me directly, and I can answer them now.
All right, great. Thank you so much, Jason. Really great presentation. And I would say too, to add to your point, you know, if people are looking to trial any of these products, feel free to get in touch with any of your extension folks and Jason, and please let us know so that we can help you set that up with you and to get you that good data. We did have a question come in through the chat box for you.
Okay.
When you were talking about the PGRs and the bactericides.
Uh-huh.
Do you know when, in relation to that freeze event, you would be applying those products? And I think we could extend that to the nutritionals as well. I know Terence is going to talk about Promalin in a minute, but did you have a sense for the bactericides or the nutritionals when you'd be wanting to put those on?
Well, I'm going to be mostly making it up, but I'll do that anyway. So for PGRs that affect phenology, you want to be the idea there is you are delaying development. So those are needing to be applied much earlier, right? You can't reverse development. So those would need to be applied prior to the stage you're trying to protect. So if you're trying to slow down bud break or bloom, you'd have to apply it beforehand. That's going to differ based on the effectiveness of the PGRs. That's also a really hard time to spray, and there's not a lot of leaves around. So the effectiveness of those come with those qualifications. For nutrient, I don't personally believe that nutrients work. And so I can't offer a good suggestion there. It certainly has to be before the freeze event. For bactericides, I don't know the exact timing, but the principle there is pretty simplistic. You are reducing your freeze risk by eliminating freezing at a warming temperature. So I'm not sure what the kill time is on the antibiotics. It's certainly something I could get back to Laura. I could look that up and get back to you. It is effective. It does work. It will only work if your ice formation at warm temperatures is the killing ice, right? So if, for example, just using numbers again, if our critical temperature in apples is -27 and ice nucleating bacteria is causing ice to form at -29, bactericides will protect your crop to -27. It will not protect it below the critical temperature of that tissue on its own, if that makes sense.
All right, thank you, Jason.
Sure.
I do want to ask one more question. I don't see any others in the chat box at the moment, but really early in your presentation, you were talking about, when a frost fan would work and, in the radiative frost versus the advective frosts.
Mhm.
And I think I personally struggled last year ahead of that freeze event where we were predicted to be a really windy kind of frost. So I thought, the wind fan wasn't going to do anything for us. And then at 11 p.m., the wind just dropped. And then I think we actually ended up having that radiative frost. But in that case, we were probably too late to really mix up that layer. I don't know if you or if Jessica's still on. Are there signs that we can look at ahead of that event where we can kind of predict what type of frost it's going to be and know when it's the best time to get the fan going?
I don't have the answer to that. I don't know the climatological part of that. Before Jessica jumps in, I do want to say one of the things that has been really illuminating for me in working with Judith is understanding that during a radiative frost, the plant tissue temperature is lower than what you would detect at the air temperature. So if you have your frost fan trigger at what you think at the air temperature, recognize the plant tissue is experiencing a colder temperature than that. So you should have the trigger warmer. Make sense?
Sure, yep.
Yep.
Yeah, I don't know if I have a great response. I guess I would say, looking just the National Weather Service forecast to see kind of what the winds are forecast to be. If you're really into looking at forecasts, you can kind of look at the fronts that are coming through and to see if it looks like it would be something where the winds are expected to continue overnight. If there's nothing going on, if it's just kind of like a high pressure system, so there's no clouds, then you would expect that radiative cooling that you could, look at some of those forecasts.
Okay, great. Thank you. We do a question from Lindsay going back to potassium, which can help reduce the freeze level. In regards to the inconsistent results, I'm wondering if the formulation could be a distinguishing factor. Do you have any experience you can share on that, Jason?
Not personal experience. I've worked with a KDL product, a single formulation of potassium on grape. But in reviewing literature, there's different types of potassium being used. I haven't seen anything where it has been shown to be consistent regardless of the formulation. Like I said, there are papers out there that show a reduction in freeze damage when the crop is in a low potassium state. So in that case, what you're doing is buffering that plant's health overall. And so healthier plants will be somewhat more successful than weak plants in surviving any stress. The other place that nutrient-based approaches work has been shown to work is in that chilling injury kind of angle. And again, that's because you're not so much surviving the freeze damage, you are buffering the plant's ability to deal with an abiotic stress. So I think it's a bit different. It doesn't mean that there will never be an effective approach. It's just that I have not found a paper and haven't done it myself to back that up.
All right, thank you, Jason.
Mm-hmm.
I also got a comment in here that Good Fruit Grower actually came out with an article recently on predicting what type of frost you're going to have and when it would make sense to put a fan on. So I'll definitely look for that and we can get that out to people as well. So with that, I think we'll go ahead and turn things over to our next speaker. Next, we're going to hear from Dr. Robinson from Cornell AgriTech, and he's going to give us some practical tips for using Promalin as a frost mitigation tool. So Terence, the floor is yours.
So our main experience with Promalin came from the 2012 year. This is a picture of little fruitlets at that year showing seed damage, but green cortex tissue. And I want to emphasize that that particular year had this kind of damage. Other years with more severe frost, the entire fruit is black. But with that background, let me show you what we found in 2012. I want to emphasize that we had two frosts. One was on April 18th at 30 degrees. This was at Geneva, but that was at tight cluster. And that's the one that Jessica pointed out earlier. We did apply Promalin 4 days after that in this particular block of 3 varieties of ginger gold, Gala, and Jonagold. But in my opinion, that frost is not the one that gave us the problems. The major frost that gave us a problem is a 3-day event on April 28th, 29th, and 30th. And those are the temperatures in Geneva. 27, I think, is where we got the most damage on the first night, then 31, and then 29 on the third night. And this was when flowers were at the late bloom stage. Some petals were falling from the king flowers. We applied Promalin on May 1st, the day after the last of the 3 frosts, but essentially 4 days after that first frost on April 28th. With the 3 varieties, we showed a significant and important effect with ginger gold at improving the fruit set, increasing the number of fruits on the tree, and increasing the yield. Fruit size was essentially the same with the Promalin treated fruits, and there was a difference in crop value. I will point out that these trees should have had a yield in the bushels per acre column of 700 bushels to the acre. And so we statistically increased it from 50 to 130 bushels, but it was nowhere near a full crop with the applications of Promalin. The second variety, Gala, there was a numeric increase in fruit set and fruit number, but it was not statistically significant. Because in this case, the untreated control had a yield of 560 bushels, and the Promalin had a yield of around 700 bushels. They should have had 1,000. So the frost reduced the yield of Gala to 50%, and the Promalin gave a numerically higher number of about 70% of a full crop, but it was not statistically significant in our statistical analysis. There was a numeric increase in crop value, but not a statistical one. They had very similar size between the untreated control fruit size and the Promalin treated. Now, the third variety was Jonagold in this block, and in this case, there was a very strong improvement in set and in fruit number per tree and in yield. These should have had a yield of 1,000 bushels to the acre. With the frost, they had a yield of 150 bushels to the acre, but Promalin increased that to 550. And so there was a really significant improvement in crop value. Fruit size was essentially the same between the treated and untreated. My explanation as to why we didn't see a bigger response in Gala is that it happened, the frost happened right at early king bloom flower petal fall. We still had a huge crop of bloom on 1-year wood, which we call lateral bloom, that was still further behind and had not yet opened. And many of those fruits survived. That's why we ended up with a 500 bushel per acre crop on Gala, because it was largely composed of fruits growing laterally on 1-year wood. The Promalin did help save some fruits growing on spurs, somewhere around 130 bushels worth of fruits on spurs, similar to what we found with the ginger gold. Therefore, we had a numerically higher yield, but although it wasn't statistically significant in this case. So that a takeaway from this is that varieties, or depending on when the frost hits, if there's still flowers that aren't open and have a slightly greater frost tolerance, and say they can take down to 27 degrees, and in this case, we only got to 27, we could have saved many of those flowers on 1-year wood. Where other varieties like ginger gold and Jonagold, which we did not have very many flowers on 1-year wood, the Promalin was the only thing that helped save some of those flowers. Now, why we had such a more dramatic saving of flowers on Jonagold versus ginger gold, I'm not exactly sure, but it shows these varietal differences. We have not really been able to test this again in Geneva. We did not do this last year with a very late frost, but I wonder whether this kind of treatment with Promalin would have any effect once you get way past bloom. But certainly in the bloom period, Promalin can have a significant positive impact on flower survival. Now, the question that comes up is, what about how other places? That particular year, we also had two other colleagues, Steve McCartney in North Carolina and Dwayne Green in Massachusetts, that did similar treatments, and we ended up publishing a joint paper. I extracted this from Steve McCartney's North Carolina data with Rome Beauty. And he has here the kind of frost that he experienced. It was earlier in April for them, April 12th and April 13th, and he applied Promalin right after the frost, the same day. So if the frost happened at 4 in the morning, he was out there at 10 a.m. spraying Promalin. And he had a significant improvement in fruit set, and this happens to be on Rome Beauty. And you can see in the next slide the kind of yield that he was able to achieve. The bushels per acre on these big trees should have been 700 or 800 bushels to the acre. The frost reduced it to 94, essentially no crop. And the Promalin, whether 1 pint or 2 pints to the acre, improved it to the high 200s, close to 300 bushels to the acre. Essentially no difference in fruit size. So these fruits that you're saving with Promalin can grow to full size. But interestingly enough, we counted seeds only on ginger gold and found the same thing, but I liked his data here, showing that the Promalin fruits tend to have very few seeds. So I interpret this by saying, okay, if we go back to the previous slide, there was 11 fruits on every tree that probably had a decent number of 4 seeds, but all the other fruits that you saved had essentially none. And so the average for the Promalin was only 1 seed per fruit. Nevertheless, the LD ratio was fairly consistent. It wasn't significantly impacted. So this means that we're producing with Promalin essentially seedless fruits. And this picture of his Rhone shows these wonderful, final, fully grown fruits with essentially no seeds. So this parthenocarpic effect from Promalin is unique, it's beneficial, and it has helped in certain situations. Now, luckily for him, he was able to repeat this on a 2014 frost event down there where he was able to test how soon after the frost do you have to apply Promalin. And so he had these 2 frosts, one to 26.6 on the 16th of April and one to 27.5 on the 17th of April. And it shows here in the bottom table when he applied Promalin. So without any Promalin, it reduced fruit set to 11%. But with Promalin, just one on the day of the frost, essentially the same day after frost, he could raise it to 20%. Waiting 1 day longer, still at 22%. Waiting 2 days longer, dropped to 16, although not significantly different. And then 4 days, and then 5 days. Waiting 5 days, he had essentially the same fruit set as 1 day after. I don't know if that's an anomaly, but this gives us the flexibility, I think, to react, hopefully on the day after the frost, but certainly you have 2, 3, 4, maybe even up to 5 days after the frost to spray Promalin and still get a response in producing parthenocarpic fruit. He did one other study, just throw it out here, whether retain could have any benefit. This one happened to be on Gala, early red one, delicious, I'm sorry. And he could show again, fruit set on the frosted trees at 44%, whereas Promalin could increase it to 55. No effect of retain. So it's fairly consistent that retain will increase fruit set in a variety of years on several different varieties, with frost occurring somewhere around bloom, and it will probably be enough to offset, or certainly offsets the cost of the product. There's not a lot we could say beyond that. We could speculate, but I summarize our results and those of others. I should mention that Dwayne Green tested Empire in Massachusetts and did not show an improvement in 2012 from Promalin. But generally, I would say that after a mild frost, and I want to emphasize that, a mild frost is where we're close to the critical temperature, slightly below it, and you get damage to the seed part of the fruit. But without the seeds, the fruit will not keep growing. But the frost, in this case, did not kill the cortex tissue of the fruit. In those cases, Promalin can cause a portion of the fruits to remain viable and continue growing all the way to harvest, even without any seeds. I'm sure that in a more severe frost, where you're way below the critical temperature and the entire tissue of the fruit is heavily damaged, there would be no way to save those fruits. Our generalized recommendation is that Promalin should be applied as soon as possible after a frost, but can have an effect even 5 days later if you can't get it on right away. The recommended rate is 1 to 2 pints of Promalin. We haven't seen a lot of difference between 1 and 2 pints. The yield improvement you get from this treatment will restore part of the crop you would have had, but you will not have a full crop. On the positive side, fruit size of these seedless fruits will be close to the size of the seeded fruit. That's what I wanted to present today, and I'll stop my presentation now and would be happy to discuss it further if there's any questions.
All right, thank you very much, Terence. Are there any questions for Terence on the use of Promalin? And Terence, just maybe you could just emphasize again. So really, it's just if you're at bloom, that's where you're really going to see an effect. If the frost occurred at pink, do you think you'd get any benefit from it, or does it need to be at that bloom timing?
No, I think it's anywhere around bloom. I would guess that anywhere between pink and 10 millimeters, you could probably get an effect. We did not test this last year where we had the late frost, and we probably would have got some better data if we had, but we didn't do it. But certainly around bloom, all the way from pink, I think, to 5, 6, 7 millimeters, you probably will have a positive effect. The reason I don't think you'll have much effect beyond that is that fruits to continue growing past petal fall have to receive a hormonal stimulus to grow. And without the seeds, you don't have that hormonal stimulus because they are the hormonal stimulus. That's why flowers that don't get pollinized, fertilized, there's no seed developing and hence no hormonal stimulus, and they just stop growing. And once a fruit stops growing, they're destined to fall off. Thus, Promalin, I think, can help to replace the source of hormone from the seed, particularly gibberellin, to keep the fruit tissue growing and keep the fruit on the tree until other sources of hormone can take over and continue its development till harvest.
All right, thank you. We do have a question from Earl. Thoughts on Promalin making that application prior to the frost versus after? Can you get it on ahead of the event?
I think you absolutely can, because the product has to be absorbed and it has to be metabolized in the plant. And so that application the day before, I think, would be probably equally effective.
All right, thank you. Any other questions on Promalin? All right, well, I think we're going to then go on to our next speaker. Next up, we're going to hear from Brent Arnoldussen. Dr. Arnoldussen is in a new position at the University of Kentucky. Prior to that, he was a postdoc in the PEC lab here at Cornell, so we're happy to welcome him back to a presentation for New York. And he's going to talk about his research on sprayable frost protectants. So, Brent, I think you should be good to go.
All right, can everybody hear me?
Yep, you sound good.
Great, okay, so thanks for having me. So I'm going to share with you today some work that I did during my PhD at Washington State University in Matthew Whiting's lab on what we call plant-based dispersions and some other things about sprayable frost protectants. Sorry, there we go. All right, so I'm just going to get right into it and talk a little bit about some of the different sprayable frost protectant materials that you might find in the literature. So Jason mentioned that you can find sprayable frost protectants back into like the 50s and 60s, and it's sort of been this holy grail kind of researchers and of course chemical companies to come up with something that growers can use. And you'll find a lot of different things. So some of them include things like low density polyethylene, some different types of foams. You can see like water-based foams, firefighting foams. One interesting one that was studied a lot in the mid 2000s at the Kearney's Research Station was kale and clay. I'd be interested to hear if anyone had any experience with that, or to know why that one never really took off. That's just interesting aside there. Some other ones are acrylic films, polyethylene glycol, and dodecyl ether of polyethylene glycol, for example. However, most of these, just never really took off because they have one or more of the following weaknesses. They're either difficult to apply, the protection that they provide wasn't very much. It had a very short duration of efficacy, so you'd have to reapply many times. Some of these were phytotoxic, which obviously, or human toxic, in the case of some of these chemicals here on the left, and the cost. So in general, if you look at these different materials, they all fall into a few different main actions. So the first is this idea of insulation, and it's been studied before. So things like low density polyethylene or these different foams, it's essentially, probably a concept you're familiar with, but it's keeping the heat in the plant so it's not dissipating in the environment, keeping that internal temperature higher. You could have nucleation inhibitors. And so this is kind of like when Jason was talking about, the ice moving around the outside of the cell. These types of materials like kale and clays or acrylic films actually prevent that ice from moving, or prevent the ice from starting to form on those different sites by blocking either the nucleation sites, or which stop it from forming, or stopping its spread. The other last is what I call cryoprotectant, or we sometimes call it antifreeze. And it's essentially like antifreeze in a car, where you put in different chemicals that go into the protoplasm of the cell, and it raises or it lowers the freezing point because of the new materials that are in there. And those are things like these different glycol polymers, like ethylene glycol, polyethylene glycol, and dodecyl ether of polyethylene glycol, which did have a good effect if you read those papers, but I think there were some issues with their phyto and human toxicities. All right, so that leads me to what I'm hoping to talk to you today, which is this class of chemical, I guess not chemicals, but yes, chemicals, but materials that we call plant-based dispersions. And so what these are is they're aqueous dispersions. So they're a water-based formula that contains nano or micro-sized particles that are made up of plant structural carbohydrates, so namely cellulose. So inside the plant cell, we have these long fibers of cellulose, and we can break them apart with different mechanisms. So some of them are breaking apart with chemical sort of digestion process, and that usually creates these crystals, or they can be broken apart in sort of like a mastication, like physical process, and that usually gives you these long fibrils. And the structure and uniformity of these particles gives them unique properties, which I'll go into a little bit more. But the most important for our purposes is that it creates this sprayable film that once that water dries, all those molecules adhere together and create this really durable pencil film. And what's really interesting about it is that it's renewable and sustainably produced from many different sources. So really, if you look in the literature, you can find basically any sort of plant waste that you can think of has been made into this. So things like sugarcane bagasse, rice hulls, soybean, you name it. I've even found things like confectionery waste, different things from different microorganisms, all can be made into this material. Most of the stuff that you're finding commercially is going to come from wood pulp and digested wood pulp. And I've listed here some of the things that I've tested previously. So the main one is this cellulose nanocrystal, and there's different levels of this stuff based on kind of its purity and how it's made. There was a really good source of this from the USDA. However, that wasn't able to make a large enough quantity of this, and that was a really expensive material, but it had some really interesting characteristics, and that comes from pulpwood. It's a nano-sized material, and it has a really interesting crystalline structure, so this is kind of what that would look like. The other ones would be something like the soybean nanocrystal. That obviously comes from agricultural waste. It's similar, but every type of cellulose or every type of plant material is going to make a little bit different shape of cellulose because of the way that cellulose is shaped. And so when you put it through the chemical digesting process, those cleaving points are going to be different. So every source material is going to make a little bit different nanocellulose. It can instead of cellulose, it could be other things like xylene, which is another it's another word for hemicellulose. That's something that we tested. That's more of a micro size, so it's not as small. It's not quite a crystal. It's more of like a globular shape. The one that's mostly commercially available now is this cellulose nanofibril that comes again from pulpwood. It's made from a massification process. It has nano and micro-sized particles mixed together, and it's more of a fibril, so it's not as crystalline and uniform. It's going to be a bunch of different shapes, and they're these longer structures. Miscanthus nanocrystal, that comes from like a biomass, like switchgrass type thing, and that again is a nanocrystal. All right, so these are all things that we had tested as part of my PhD. And so why do we think this was going to be a good frost protectant? So before we started, really looking at the mechanisms in plants, we saw two things. First was that when it dries, it creates this film. Within minutes, this was something we sprayed in a pre-treated plate, it dries, and within minutes you're left with this film that you can actually pick up, and it's like this tinsel strong film that's left from when those molecules sort of bind together as the water evaporates. And when we tested its thermal conductivity or its insulative potential, we found that it had half the thermal conductivity of the next best insulated material that was tested in literature. So it's a really good insulator for its size. So taking that a step further, we did some scanning electron microscopy scans of this stuff. We looked at the thickness of it, and you can see, this is a sort of a side sectional view of it. You can see it's a very small film, so just about 30 to 40 microns. And what's interesting is that the more you can concentrate the material, the thicker your films are going to be, which of course is going to make it more effective. But there is a limit to that. And that's something that probably material scientists need to figure out. But once you get, with some of the materials, or once you get past like a 3 or 4%, this material becomes too viscous and can't be sprayed. So it's balancing sort of the thickness of the liquid so that you can spray it and getting as thick of a film as possible. And you can see here from these photos on the right, the upper photos being at a lower magnification. You can see it's not very clear at this lower magnification, but hopefully you can tell sort of the change in texture. So on the right is the treated, and you can sort of see the films just by the change in texture, and it becomes more apparent at the higher magnifications, where you can see sort of the texture of the bud surface, whereas you can see now there's clearly a film on that bud. All right, so to test the insulative kind of hypothesis and the insulative effect, we did some controlled freezing and thermography, kind of similar to the video that Jason showed, but we were looking at a different parameter. So instead of looking at temperature, we're looking at something called emissivity. And emissivity is just the ability for a material to radiate energy. And what we found is that, so you can see here on the right, the CNC treated. So the lighter the color, the higher the emissivity. So the higher the emissivity, the more energy can pass through it. So the buds that were treated with CNC are darker, therefore they're letting less energy out. So the CNC coated cherry buds on average had an emissivity that decreased about 16%. So we think there's more to the story, so we did some more tests, and what we did was something called calorimetry. So if you've heard of differential thermal analysis, it's sort of similar to that. It follows a similar principle, but whereas DTA is sort of this sort of high throughput way of telling cold hardiness status of buds, this was more to look at both the water and thermodynamic status of the buds when treated with nanocellulose. So We cooled the buds to about -15°C. We cooled it at a much faster rate than you would like to if you're doing, say, DTA, but that's just because of the limitations of the machine. And we used application rates of 0, 2, and 3% and looked at them 1, 2, and 3 days post-application. And what we look for, these are the sort of freezing events that sort of coincide with kind of the extracellular, intracellular freezing like Jason was talking about in his talk. So right here, this is what we call the HTE. This is this large freezing event, which represents that extracellular freezing, which is generally considered, a non-lethal freezing event. Then we have this event here, which is the LTE, which we consider a lethal freezing event. And then what we were looking at is something called the enthalpy, or the free water. So this is what we call supercooled water. So this peak essentially represents the remainder of the supercooled water freezing, and we can actually measure that to figure out actually how much water was left in the cells at the time of freezing. All right, so to look at the exotherms, what we see is that both the HTEs and the LTEs are significantly affected by both the dispersion and the concentration and the time. So we found that we see that actually pretty consistently when testing CNC, particularly on dormant buds, is that the effect actually goes up with time, which is very interesting. And that was on both the HTE and the LTE. So it showed that isonucleation generally is being inhibited by these dispersions. When we look at the enthalpy results, again, we see the same effect with time, which was really interesting. But what we're seeing is that the CNC films are actually retaining more energy and most likely water during cooling. So what that leads us to believe is that likely what's happening is that for some reason we're having more supercooled water in that, which likely means that there's some type of nucleation inhibition going on, which makes a lot of sense considering the nature of this material. It's a film, which probably just like the kaolin clay or acrylic films are blocking those nucleation sites or preventing the ice from moving up the plant. All right, so I want to shift gears a little bit away from the mechanisms of this material and talk about just looking at different materials. So I'm just going to show a little excerpt of some of the different materials that we trialed. This was a trial that we did where we looked at miscanthus, that was that biomass grass material, and the CNF, which is most similar to the commercially available product that you can get now, or is in development now. And we tested it 1 day, 3 days, and 6 days post-application. And what we find is that there is actually statistically a different effect from both the control, the miscanthus CNC, and the CNF. Additionally, we see that actually changes to where by day 6, while both are still effective, we're seeing as much as a 6 degree, almost a 6 degree difference in the change in LT10s. While that's still effective, the miscanthus seems to be less effective. So all that to say that different materials are more or less effective and have more or less durability and a range of efficacy. All right, so while that all seems really good, keep in mind that a lot of that was on dormant stuff that we could use DTA on. So what happens when we start to look at buds that are deacclimating and starting to move towards flowering, where frost damage is more problematic? So it starts to change. So this was a trial that I did where I looked at different bud stages. I looked at early bud stages and later bud stages. So these are in cherries. So I looked at 1 day and 2 days post-application, and these were all tested with a controlled freezing test. So that's essentially where we take a subset of samples and we expose them to a gradient temperature and then analyze them visually for either live or dead. So that's what these zeros or ones represent here. So as you can see, hopefully, is that at these early stages we get really good efficacy, and we even see that a similar effect where that efficacy is actually increasing over time. we see that they're starting to become a little bit less effective as we get to first white, but still statistically significant. However, I want to point out that as we get into these later bud stages where we're probably going to need them, they actually become both less effective, and the duration is a lot less. So that is likely because of the fact that we have less that can be encased. So when we have the small buds, they can be encased by this film and protect it, whereas there's really nothing that can be well cased here on open flower. So as we get towards full bloom, we see no effect at all for this, and that really I think kind of limits the potential of these as we likely are going to have a lot of frost events at full bloom. However, I would like to point out that we haven't tested on things that like petal fall or small fruitlets, which I think that might actually see the efficacy be reestablished again as we get to that point. So that could be something interesting to trial in the future. All right, so in that same trial where I looked at all these different stages and we're applying onto open flowers, I also looked at if there was any secondary effects. So I tracked bloom rate to see if this had any effect on floral development, and I didn't see any effect on both floral development or fruit set. I think it's important to know that if we were applying some of these to our crops, if we would have any secondary effects of that. I didn't see any. All right, so to wrap up here, is this a new tool that could mitigate cold damage? And to look at that, I want to look at some of the disadvantages that I mentioned in the past and see how these plant-based dispersions kind of stack up. So I mentioned that a lot of these old materials are difficult to apply. We've actually trialed this material with electrostatic sprayers and typical air blast sprayers and tower sprayers. So a lot of your typical orchard application equipment can apply this material. We found that has a really good degree of protection with a single app, can protect up to 6 degrees Celsius. Short duration, so this can last up to 7 days, but there's a huge caveat there, and that is that it has to be dry. So, and that might be an issue for sort of the eastern U.S., whereas out west, we can have frost events and it can still be very dry. It might be different here. So we saw any sort of dew or any like melting snow or any moisture issues, and that stuff, the same characteristics that make it able to be, dissolved and sprayed means that it will dissipate and wash away immediately with any sort of moisture. So we also saw that there were some issues at the later stages of duration when flowers are going to be most vulnerable, but we didn't see any phytotoxic events. And that's sort of leads us to the last thing, which is idea of cost. And that kind of brings me to this idea of the commercial availability of this product. So this was licensed, the IP was licensed in 2022 to a company called Sappi, which is they're the world's largest like wood pulp digester, and then they make different nanomaterial products for a bunch of different industrial and packaging type applications. And I don't exactly know where the state of this is, so it's been a while since I've worked on this, but I know that this is a great company that should be able to put a lot of R&D behind this product and hopefully make this available in the future. All right, so I think that's it for me. So I'm open to take any questions. So if you have any questions, use my email. If you had any questions specifically about the material or where to get it, I would forward those to Dr. Whiting, and I have my email here and his below. So, all right, with that, I should be open to questions if you have them.
All right, great. Thank you so much, Brent. Really interesting presentation. We do still have 6 minutes till 1:30, so please do send in your questions for Brent and for the group at large. Brent,
Hey,
we do
Mike,
have a
it's
question from
Dan.
Philip.
Can I pop in with a quick question?
Yep, sure. Go ahead, Dan.
Yeah, okay. Brent, great presentation, really informative. And you mentioned there was no effect observed on fruit set. But I'm curious,
Yes.
if the application timing was at, say, full bloom when all the flowers are actually open, so the parts inside are sprayed, by the material,
Yes.
could there be an effect on pollination?
That was actually exactly what I tested. Let me go back to that. So what I tested was I sprayed it at different timing applications. So I tested it at first white, first open, and full open. So I went out right at bloom and then sprayed on there. I even looked back to see if there was still like foraging activity, which I didn't take data on, anecdotally look at that. But I didn't see any effect on that or any effect on fruit set in those blocks.
That's great. Thank you.
Does that answer your question?
All right, great. Thank you, Dan. We have a question. Any comments on kelp-based products? Brent, I don't know if you've worked with any of those or anybody else that
No.
presented today have any experience with those?
We're testing some stuff with kelp-based products in grape. I think a lot of these natural extracts are going to have a lot of natural PGRs in them, as well as nutrient mixes. And so the challenge will be, if you see an effect, if you can disentangle, I mean, from the grower's point of view, if it works, it works. But from the science point of view, when you have a cocktail, it'll be tricky to figure out the mechanistic angle on those, and that will tie into the consistency across cultivars and regions. But it is something that, yeah, we have in the hopper.
All right, great. Thanks, Jason. Any other questions for any of the presenters today? If not, I would probably ask the group. I mean, considering everything that we heard today, perhaps just a summation of, moving forward, what do we want to do? It sounds to me like, keeping an eye on our weather forecast, using those different climate tools that are out there should be first and foremost, so we can stay ahead of the weather predictions if we do have any of these frost events. Making sure the wind machines are ready to go, as Jason mentioned. They seem to be one of our main sources of being able to give us some mitigation. It sounds like from what Brent was talking about with some of those new materials with those nanocrystallines, when they're commercially available, sort of at that earlier bud stages, they might be a potential for us. Is that accurate, Brent? Have you tried that on apple? I was curious about that. It sounded like a lot of the work was on sweet cherry.
A lot of the work was on sweet cherry, but we did, so that first paper that I mentioned was an apple, and I did have something that I published in a conference paper on apple. So we did do work in apple, but mostly it was focused on sweet cherry there. And yes, it seems to be most effective, kind of at dormancy, eco-dormancy, and those earlier stages of bud development. Sort of, I think pink is kind of the limit. Once you're spraying that pink, it's not going to be effective as long. I do think it'd be really interesting in apple, and I think our needs are a little different here out east, so we might want to look at it sort of at petal fall or those later, like smaller stages of fruit development, because I think it actually could be effective there. We just haven't tested that.
Okay, great. Thank you, Brent. And then once you hit pink, as Terence was telling us, maybe thinking about switching to Promalin if your frost event occurs around then, so you can set those parthenocarpic fruits. Is sort of the strategy that I get the sense of from this meeting. Just wanted to see if anybody else had any other thoughts on that or other suggestions.
Mike, I don't have any more suggestions, but I do want to give my opinion that I don't think there's evidence right now of a serious frost event coming up in the next half of this month. I think that's a little bit of premature scare tactic, and I don't see it in any of the forecasting that Jessica talked about or any of the other weather forecasting up to the end of April. So it may come, but I don't see the evidence of it yet.
Certainly, yeah, and fingers crossed that we don't.
Yeah, I can just, continue on that, just, based on, what I had shown there, the possibility is there, but we don't have like an event forecasted. So just kind of, I guess, stay tuned and keep watching.
All right, thank you.
Jessica, could be possible that through the extension channels, we could update the 8 to 14 day temperature outlooks. with your forecast as we get closer to bloom? Jessica?
Yeah, the 8 to 14 day are great to look at. And also, I mean, once you get the National Weather Service can put out about a 5 to 7 day forecast. So you can, you can keep watching that as well. So I think, yeah, those are both good resources to look at.
Thank you for a collaboration. As we get closer to bloom, perhaps we can update the industry and you can help a little bit so we can use those graphics.
Sure.
So we can send that to the growers.
Hey, Mike. I have a question about retain as a protectant, and I wanted to punt it over to Terence since I haven't worked with it.
Certainly.
Well, I didn't see the question, Jason, but
It was, can you discuss any effectiveness of retain on frost protection? Yeah,
Yeah,
it came through direct message.
the best data came from Steve McCartney in North Carolina, found no benefit of retain applied after a frost. Now, retain is an interesting product in that it takes typically about 5, 6 days to really have a metabolic effect on the plant. That's why in pre-harvest drop, you have to have it on a week or two before you drop. So I'm not sure what it would do before bloom or before the frost event, but it didn't work after the frost event.
All right, thank you, Terence. All right, not seeing any other questions. If there's no more discussion, I think we'll go ahead and close out for the day. I'd like to thank all of our presenters for joining us today, all my colleagues helping set up this meeting, and for everybody attending. And certainly we'll keep an eye on the forecast and hope that we don't end up having any of these freeze events. So fingers crossed, and hope to see you all on the other side of this spring. So thank you.